This will be a video reviewing the stages of mitosis. We'll start with prophase because it's the first one. You can see a pretty good image of that over here on the slide, showing how what the cell actually looks like. Got a bunch of interphases kind of surrounding it. A good anaphase right there, by the way, as well. So what actually happens? The nucleolus, which is inside the cell, nucleus will start to disintegrate. The chromosomes are going to start to be seen. We get the chromatins out of that, all those loose fibers, and into bundles, and the membrane will start to break up and disintegrate. You can kind of see that a little bit. It's harder to see. An event that you can't see in an image is that the centrioles will split and move to the opposite poles or ends of the cell. Now that only occurs in animal cells, so that's why we won't see it in this cell. And even in animal cells, it's kind of hard to see. You might also start to initially see the spindle fibers starting to form as well during prophase. Another image of prophase, this one's kind of a little bit more zoomed in. Definitely can see the chromosomes. You can see the membrane starting to break apart at times. Okay. The mitotic apparatus will start to form. You get these asters, which will kind of give it appearance. Now, the drawing I'm using is a plant cell, so you won't obviously see it, but you start seeing these things. I always think of those like head scratchers, what they kind of look like. The origin right there is a centriole right there, so they'll start to appear as well. And the chromosomes have doubled, remember, in interphase, so they'll have a double amount that'll be visible during this time. The actual spindle fibers coming off here, those will start to attach to each individual chromosome, kind of as I drew it. So that's all happening in our prophase. Some books will divide prophase into two stages called prophase and prometaphase. For simplicity, we just keep it as one. Here's another just diagram showing that occurring. Here's the centrioles moving to the opposite ends of the pole. This makes this have to be an animal cell. You should know that. It's an animal cell because of that. And then the nuclear membrane's breaking apart. This part right here on the chromosome, so these two are identical to each other, that's called the centromere. That is where that microtubule will attach to the centromere. Just showing it so far, comparing it to interphase. Notice interphase, a big mass. You can actually start to see the chromosomes present there as well. Definitely can see them here. The membrane's really broken apart. These are animal cells, by the way. Good image of comparing interphase to prophase. Now in metaphase, a lot of times guys are confused on what's happening here, so I'm going to walk you through it. Here's one individual cell. The centrioles will be right here and here. The spindle fibers are this part right where I'm kind of going with the green, right there. The chromosomes are these darkly stained things in the middle, right there. A lot of times people will see this and think that this is anaphase because of this pink area here. It's the darkly stained, it's the chromosomes. They are lining up in the middle. During metaphase, the chromosomes, partners, line up in the middle. Okay? And what ends up happening is they line up. If this is one chromosome, this is an identical chromosome. Okay, should be identical, bad drawing. There's our centromere, our microtubule attach here, but these are both the same identical information on those. Okay, that way when they get pulled by this setup right here, each new cell will get the same number of chromosomes with the same exact genetic information. So it's a way to ensure the information is parceled out equal. Once again, what's important is that how this lining up and hooking up occurs happens properly. If it doesn't, we're going to have some issues. Just showing metaphase in different images. So in a plant, there's the equatorial plane, the middle of the plant cell. You can see them lining up in an animal cell, a plant cell, and then here's just another diagram. Okay, there's our centrioles. Those are the microtubules attaching, pulling apart the chromosomes. Notice those are two chromosomes. There's one, there's two, being pulled in opposite directions by the microtubules. So anaphase is the third stage of mitosis, and in this case the centromeres will split, causing the sister chromatids to separate. So when we look at an actual diagram of a cell, you have, there's a chromatin, okay, there's a chromatin. Now these are supposed to be once again identical, so therefore we call those sister chromatids, okay? So that's one sister, that's the other. Right there, okay, that is where the centromere will be attached. There's our attachment. Microtubules will attach at those locations and then pull in opposite directions, causing them to split if this is happening properly. All right. During this phase, we will also get the start of cytokinesis. So cytokinesis happens during anaphase as the cell's being split. Remember, cytokinesis is just the division of the material in the cell. So the cytoplasm, the membrane, all those other things. Not as important to get exactly right as when we divide up our DNA. Got to make sure our DNA is divided up equally. In plant cells, what they do is they do a little bit different approach. If you think of cytokinesis in a plant cell, they kind of build it like by a brick. So they just keep adding the material, and it keeps going back and forth on that cell plate, and eventually more of it's added, more of it's added, more of it's added, and then we get and somehow I jumped around. I apologize for that. Animal cell, we get this cleavage furrow where what ends up happening is that, think of like elastic on a sweatpants or whatever. It's tightening around this area here, causing them to separate. I'll kind of show you that visually. It's a little bit easier to understand versus my diagram. Bunch of cells that you can kind of see an anaphase here. Good example of an anaphase. One way you can always kind of tell, it's these fingers. Looks like fingers getting pulled apart from each other. That's a good way to tell anaphase is occurring. And then there's another one of anaphase as well. A wall dividing it. So in an animal cell, which this one would be, you'd start to get some pinching effect here. Eventually what we're going to do is the tightening here causes those to cleave off into two different cells. So telophase is the fourth and final stage. Basically, if you read through this part, it's really just the opposite of prophase. It's all the opposite. Now, in this case, the nuclear membrane reforms, where during prophase it fell apart. The nucleolus reappears. Remember, before it was disintegrating. The chromatin unwind versus they were winding up. So all these stages really mean we're the opposite of prophase. How do I know the difference? If I were to look on a slide, well, that's easy. If I see two cells that are right next to each other that are both look like they're in prophase, well, they're not. What it is they just got done with telophase. And so that's a good way to tell the difference. I'll kind of show you that on the next diagram here. What's the result of mitosis is that we get two daughter cells that are identical to each other, and that would have been identical to the original parent cell. That's if everything happened properly. So when this is occurring, we get identical copies. Great images of telophase occurring. This is in a plant cell up here. You can see the cell plate is already formed. This is late telophase. It's pretty close to being done. Here's an animal cell. There's that cleavage furrow where we're dividing the two cells. This is going to be some proteins will be in here, creating the tight band that'll separate the two cells. Another one here. This one's a little bit earlier. Notice there's still a little bit of a wedge or opening right there between them, but this is definitely a telophase. And then last but not least is the diagram. Another image just showing this occurring. I like this diagram, kind of has all the different stages, kind of mapped out exactly what's going on. What they do that's a little bit different from what we do is that they're going to have two stages here. We just combine those into one called prophase. But it has all the different processes, two daughter cells that are produced. So you can kind of read through these, make sure you understand and identify those different parts. I'll leave this on for a half second. And then this is a good one to kind of test yourself here to be able to identify all the different cells and different stages. So like right here, this cell right here, if you're asking me, I would say that is in late anaphase or early telophase. I probably wouldn't ask that on the test just because they're so close. I have better examples on this one. But if you're wondering, that's where I kind of see it. What do you think this one is? Oops, up there. Definitely a metaphase. They're lining up in the middle on the equatorial plane. That one. That is a anaphase. They're being pulled apart in opposite directions. That's a telophase, anaphase, anaphase. There's some metaphase being lined up. That's a late telophase right there. Bunch of interphases. Those are a bunch of interphases right there. Interphase, interphase, interphase. So notice a lot of interphases. Anaphase, metaphase, prophase, prophase, Okay, prophase, prophase. Anaphase, either being pulled apart. You should be able to take any of these images that I give you, that's an anaphase, these are interphase, and be able to identify them based on their stages. That's what our lab will be doing in class today. Thanks for watching the video. Hopefully it helped. Have a good day.